All right, outside of class. Today, we're going to talk about the neuron. We're not going to go into depth about the anatomy. We'll do that in lab, but I'm going to give you a little bit of this now. 4 general regions of a neuron. We have the dendrites. This is basically where, so a neuron in general is an information processing cell. It's a unit that we use to process information. So the information has to come in somehow, and it comes in through these dendrites. All right, that's the first general region of a neuron. The second region is the cell body, or soma. And it's broken up into 2 very gross categories, structural categories. First is the nucleus, which is where all the genetic material resides, and then what's called the perikaryon. The perikaryon, which is, karyon just means nucleus, essentially, the origin of the cell, and then peri is just nearby it. So it's this cytoplasm, the neuroplasm, that's near the cell body, the perikaryon. I used to make a joke about Rick Perry and these. When I taught this in Texas, there was this thing about how he had left all these, what were they called, coyotes on the side of the road, dead on a hunting trip that he went on. So, but I guess that joke doesn't really hold any longer. He's now the Department of Energy chair, isn't he? The axon is the third region. And that's broken up into the axon hillock and the axon proper. The axon hillock is a special region that you'll see is shaped a little bit like a funnel leading in to the axon. Hillock means small hill. It's an important region because this is essentially the trigger that causes an action potential to propagate down along the axon towards the fourth region, the telodendria, which is the neuronal terminal. So this is where outgoing signals are passed on. So incoming signals, processing, and outgoing post-processing signals. All right. So there are 5 basic membrane processes that happen in any neural activity. And we're going to go through each of them so you can understand exactly how a neuron works. The first concept to have is basically how a cell establishes its resting transmembrane potential. It's V sub R. That's the symbol we're going to use. So V means potential. What does potential mean? What is potential? Tell me what that word means for you. Don't presuppose what I want you to tell me. Just tell me what that word means to you. What is your potential? Somebody, tell me about your potential. When they say you have such great potential, what does that mean to you?
You can go places. You do stuff.
You can do stuff.
Yeah.
You can do stuff. It doesn't mean you are doing something right now, right? It means you can do something. There's the potential for change. There's the potential for action. So I am up here. I have I've stored energy in my body. My muscles put me up here. There's now energy in my body, and I have the potential to release that energy. I could just spend the rest of my life up here, or I could release that potential energy that I've stored in my body by jumping down here. Okay? And that energy got released by the force I put into the ground, the sound, etc. You can have electrical potentials. Most of you probably took physics in high school and have heard all this before. But an electrical potential is the ability of any system to have, it has stored energy that can lead to some sort of change. All right? It can lead to some sort of action. That electrical energy can be released in one form or another. And so there is a resting transmembrane potential. It's kind of like a mousetrap that's been set, right? There's this potential energy stored in it if we can just get it to release. All right. The graded potential. So a graded potential is a change in the transmembrane potential. It is temporary, it is localized, and something causes this stimulus. The way I think about this is a graded potential. I hope I'm not going to be too crude with these examples, but graded potentials have different strengths, right? They have different strengths. And that's why they're called graded. They have certain intensity which falls away from the center point of the initial stimulus. So for example, I could sit up here while lecturing and just let out a big belch. I could just belch in the middle of class. Horrid, right? And you may all think yourself, like that. Oh, God, Dr. Klepak just belched in class. And you may, go back to your dorm suites and tell your suite mate about how you have this crude professor that did this. But it wouldn't go that much further than that. It causes a stir in the class. Golly, Dr. Klepak. And you tell the story, but it doesn't go beyond that, right? Or perhaps, I don't know, a fireman could save a bald eagle who got trapped come up with some sort of story that would make it into the local newspaper, right? It's an event that happened that the stimulus spread out a little bit further. Or you could have, an alien land in the middle of campus, and it would be a stronger stimulus, and the news of that would spread out and ripple further. This is a graded potential, all right? It's going to cause a change in the membrane potential that is going to decrease with proximity towards the point of original stimulus, all right? Does that make sense? We're going to go through it in much more detail if those analogies don't help you. Action potential. It is also a change in the transmembrane potential. It's well characterized as an electrical impulse. It's triggered by a summation of graded potentials. It's triggered by them. All right? It is not the sum of graded potentials, but it's triggered by the sum of graded potentials. And we'll see what I mean by that in a bit. What it does is it propagates along the surface of an axon. They haven't replaced this bulb yet. So we have resting potentials in these membranes. We have signals that come in that disturb this resting potential and cause graded potentials that dissipate from the point of stimulus, wherever that may be out here in these dendrites. But if we have lots of inputs, those graded potentials add up and trigger something at this axon hillock, which then causes this mousetrap to spring, and we have an action potential that gets sent down the length of the axon. So this action potential then propagates itself to the synapse in the telodendria, down to the synaptic boutons. Traditionally they're called synaptic boutons or buttons. Bouton is a French word, but they also are called synaptic knobs now. So this action potential comes down to the synaptic knobs, and we have synaptic activity. A synapse is simply the gap between a presynaptic bouton and the postsynaptic terminal on some other dendrite of some other neuron. All right? So we're going to send the signal from here to here across this synaptic cleft. A synapse is just a gap. All right? So there's this synaptic activity, and we'll talk about exactly what happens in a synapse. And then lastly, we actually could, I put it 5, but we could put it sort of back at 3 in the list here. It's this notion of information processing. So exactly how is a neuron processing the information it's getting in? The information as represented by different signals that are coming in through the dendrites. How is it organizing those into some sort of meaningful output? Okay? So this is many people's careers, whole health science industries span the different parts of this, and we're going to fit it into 1 hour-and-a-half lecture here. So we're going to gloss over some of it, but I'm going to try to get some mechanics down for you. All right. Any excitable tissue, and there's 2 types of excitable tissue. There's nerves and muscle. So and together they're called neuromuscular tissue or excitable tissue. Depends on whatever dynamics we're able to describe in this cell membrane potential. It's by manipulating this cell potential that these cells are able to establish that any excitable tissue is able to do its function. Right here we see some smooth muscle cells, which are giant, and they have, this is the motor end plate full of a bunch of synaptic boutons. This is a motor nerve. These are all the telodendria. And this could be a little slice out of some muscle somewhere. All right, so this is showing the breadth of neuromuscular tissue. So early micrographs show, so this is a freeze fracture. This is freeze fracture electron microscopy. What they do is they take a sample, they freeze it, and then they fracture it. They basically crack it. And what happens is sometimes those cracks lie along the plasma membrane. So it rips the top half of the plasma membrane out, and it leaves the bottom half. So here would be plasma membrane, the lipid bilayer, and here this top layer of the plasma membrane has been ripped off, and the bottom layer is still in place, but it reveals all these transmembrane proteins that are embedded in that membrane. Okay? It's these proteins which are going to help give a membrane its special electrical properties. Okay? I'm going to anticipate it a little bit here by saying that it's through the selective sequestration and passage of various charge carrying species. So ions, cations, anions. All right. In a lipid bilayer that has no proteins in it, things don't pass very easily. Steroid hormones do because they're lipophilic. They can dissolve into lipid. But, water doesn't pass very well. Solutes don't pass very well, like sugars or proteins or ions, electrolytes of any sort. So to enable this, we have these proteins that get embedded in the membrane. And it renders the cell membrane semipermeable. Not completely permeable, but semipermeable due to the action of these 2 different types of doorways, we'll call them. Channels and carriers. So a channel is just a protein that's a pore that's going to allow solutes to pass. So for example, this would be a channel. It's just a little hole. It's just a little doorway. All right, like this one here would be a channel out of this classroom. You could just walk out, whatever. I'll walk in. And a carrier is something that actively pushes whatever it's supposed to be regulating through and doesn't just allow the free passage of solutes across the membrane. So this would be like putting a bouncer at the door who would control who's able to come in and out. All right. There are two factors that are going to dictate what's passing through a channel. Concentration gradients and channel conductance or gating. So a concentration gradient, for example, say the word got out that I just had the most engrossing, spectacular, fantastic class. I was giving out $1,000 bills at the end of my lecture, and people were lining up to hear the words of wisdom and be endowed with all that, right? Let me just continue to flatter myself. The class, the hallway would be packed with people, right, out there. It'd be packed with people. And they would be pushing to get in, all right? So there would be this gradient. This gradient would be established. Right now, there's a lot of room in here. There's a ton of people outside. And so people will be going down the concentration gradient. There's room in here, not room out there. People would be coming in. Until the density of people in here equaled the density of people out there, and it would sort of level off. The second thing that is going to dictate the movement through channels is the channel conductance or gating. All right, so how do we gate a channel? We have these three different examples of how you can gate something. You can have a chemically gated channel. So here's some sort of ion that wants to come through this channel, and acetylcholine can't get through. Acetylcholine, which is this other chemical, it's this chemical that acts as the key. It comes in, binds, and then finally these ions are able to pass through that channel. We can have electrically gated or voltage gated, potential gated channels. This here is just a voltmeter. It's just like a battery tester that's testing, it's measuring the potential across the membrane. So it's called a patch clamp voltmeter. And we see here at negative 70 millivolts, this door on the gate is closed. This one is open. However we're going to do it, we're going to change this potential, and when that potential change, transmembrane potential changes, this door swings open. It swings open, allowing free passage. But it continues, when this happens, the passage of these ions is going to further exacerbate the change in the transmembrane potential. This door is going to swing shut. This is the inactivation gate. Not all channels have two gates, but I'm showing you an example that does. Okay, some kind of voltage gating. Then there are mechanically gated channels, and we'll see those. Those tend to be used in various sensory receptor neurons. So in your ear or in the touch, deep touch, when you get the tactile sensations, you are deforming the plasma membrane of the sensory neurons and opening those channels because of that. All right? All right. And any of these examples can have three various states. They can be closed but capable of opening. So, for example, here, this is closed, but it's capable of opening. We just have to change the transmembrane potential. It can be open or activated, or it can be closed and not capable of opening. Here, it's inactivated. This is the inactivation door. If we want to open this channel again, we have to somehow reestablish this transmembrane potential that we had at negative 70 millivolts to be able to pop that door open again. All right? So you can either be closed but able to open, or closed and not able to open. Carrier proteins. So these are proteins that somehow are going to interact with the molecule that's being moved. That means, for example, you could have a bouncer that just opens the door and lets people in. Doesn't interact. It's just, somebody, a doorman who has the door open and somebody is just coming through. Or you could be at some sort of royal gala and you could have an escort that has to walk every person in. They're physically interacting with each person. It's going to limit how fast people can come in, right? That means it's saturatable. You can put a zillion people outside, but they can only come in at a certain rate because that carrier protein is actually interacting with each solute that's passing through the door. It can get saturated. All right? There's a maximum kinetic threshold that the channel has. So this transport with carrier proteins can either be passive. A passive one is one where it's either just diffusion, it comes through without a carrier protein, and this is only a small class of molecules that are going to be able to do this, or it can be, it can just come through because it's a hole and it's going down a gradient. There's more outside than there are inside, all right? Or it can be some sort of facilitated diffusion where it is, this is saturatable here. This is not really very saturatable in the same way this is. It's only allowing one through at a time, still being driven by its gradient, okay? Or you can have active transport. So there's not very many out here. There's a lot in here. And so it's taking energy to stuff more people in here. These molecules don't really want to go in here because there's too many people in here already. And it's taking energy. It requires an input of energy to make the thermodynamics of that possible. Okay? It's thermodynamically disfavored to stuff more in there. And so, because it's basically the entropic term of any thermodynamic expression, if that makes sense to you. Because by concentrating something, you're imposing order. You're opposing the natural tendency for the universe to disorder itself. And so if you're paying that entropic cost by concentrating something, sending something up its concentration gradient, then you got to pay for it in terms of enthalpy or energy, that the heat of energy that you're putting into the system. That make some sense there? All right. Here are some more ways of distinguishing carrier proteins. We can have a uniporter. It's something that just allows a solute to go in one direction. Its job is just to allow, it's just the indoor or the outdoor, but it only is one way. We can have antiporters. These are carrier proteins, channels that allow something to go in one direction only if something else goes in the other direction. Okay? It's like going to a club where they're at max capacity, or like the pool. Go to the city pool. It's something from my actual life. go to the city pool on a Sunday in a January, and it's cold outside, and everyone wants to jump into the 94-degree pool at the YMCA, but the pool is at capacity, and you got to stand there waiting with your towel and your little kid who wants to go into the pool until somebody comes out. So for every person that comes out of the pool, somebody can go into the pool. It's an anti-porter, all right? Or there are sim porters. It's a doorway that only allows something to come in if it comes in along with some other thing that it's supposed to be coming in with. All right? So you can imagine by manipulating all of these different permutations, it's quite a broad toolkit that any particular cell has to establish some unique membrane potential dynamics. By the way, I don't do this for all the lectures, but in the early lectures, I put the references to the digital texts that I have down in the corner. So if you want to read more about it, you can do that. All right. Electrical gradients. So there are some passive forces acting across the plasma membrane. Here's a big protein, and it turns out that the surface of most proteins is kind of acidic. It's kind of acidic. There's not a lot of basic proteins. There's not as many basic amino acids. So most of those amino acids are kind of acidic. And because they're acidic, at the reduction potential that is the cell, most of these acidic amino acids are actually in their conjugate base form, meaning they're ionized. They've already given up their proton, so they're negatively charged. All right? Negatively charged. These proteins are big. Often, even if they're a single protein, they're big, or many of them are these macromolecular proteins, part of a cytoskeleton, or who knows what. They're big, and they can't get out. They're stuck in there. They can't get out of, they can't pass through these channels. All right? And because of that, all of this sets up this arrangement where if we have a transporter that is pumping positive ions out, it's trapping all the negative charge inside. We have a transporter that's pumping positive ions out of the cell. The negative charge can't follow it. It's stuck in there. This is how we turn it into a battery. This is how we turn a cell into a battery. We're separating charge. That's all a battery is. That's all a potential is a separation of charge. It's a separation of my mass from the earth if I climbed up here. All right, that's a potential. Separation of water at the top of the waterfall from the bottom. Is this making sense? Pretty fundamental stuff. All right. So we've talked about potential. We've all got V equals IR. Ohm's law. Potential. The potential for work, the potential for energy release of any system is simply the product of the amount of current that system sees times the inherent resistance to that current. All right? That's the potential to do work. So for example, if this room was full of Jell-O, and I climbed up on this table again. I'm not going to do it again. But this room is full of Jell-O. I climb up here, and I jump off. I have a smaller potential than I would if I climbed up here and jumped off in the air. The current is the same. The current is one human jumping off. but there is a higher resistance because my body is being opposed by the jello that I'm supposedly sinking down through. All right? In electrical terms, electrical current is simply the movement of charge. The movement of charge to dissipate charge separation or potential difference. All right? That's current. It's just the movement of charge to dissipate, to short circuit that separation of charge that we've imposed on a system. And resistance is just how much the system is resisting that flow. What do the charge carriers need to work against? All right, so the amount of current a membrane restricts in this example. So here we have some sort of potential difference, and this is a stream, and this is how high it is from the top of the stream to the bottom of the stream, and the water flows down and gives us a certain current. To get that same current in a stream that has all these rocks in it, and that's resisting flow, this is actually not a very good example, but you have to increase that potential difference because we have higher resistance to flow than in this one. If you want the same current output, you have to increase that potential difference. Is that making some sense? All right. So that's a potential gradient, like an electrical gradient that we just talked about. There are chemical gradients too. And what I'm moving you towards is the combination of an electrical gradient and a chemical gradient. We're going to call it the electrochemical gradient. So a chemical gradient is just a concentration gradient. It is not a charge gradient. It's not a charge gradient. It's just we have 3 different color blocks on one side and 3 different color blocks on the other side. Are they equally distributed or not? So here we have these green balls, which are the chloride ions, and there's 4 of them per volume on this side, and there's only 2 per volume on this side. So the chemical gradient wants to drive the chloride this way across the membrane. There's just more of them out here than over here. That's entropically disfavored, that we've imposed artificial order on the system. Do people get that concept? Does anybody not understand that concept? Okay. And then likewise for the sodium. There's more sodium out here than in here. So sodium has this chemical gradient in this cartoon driving it this way. However, on the other hand, we have potassium. There's more potassium inside than outside, so potassium wants to go that way to level itself out. Has nothing to do with charge. Has everything to do with concentration, chemical concentration gradients. Okay? It's just like you take a glass of water, a big beaker of water, a tank of water, you drop a drop of food coloring in it, and with time, that food color, in the beginning, it's all concentrated right in one spot, but with enough time, all those dye molecules spread out evenly. Nature abhors a vacuum. Anything that has been concentrated in one place requires energy to maintain that concentration. Otherwise, it's just going to diffuse out. This is the concept of a chemical gradient. All right. We've talked about how the resting potential is the transmembrane potential of an undisturbed cell. And that number in... Oh, yeah.
Yeah, sorry. On the previous slide, at the bottom, it said VTM. What does that stand for?
Yeah. VTM is the transmembrane potential.
Okay.
Yeah. I kind of skipped defining that. It was on one of the other slides. But VTM is different than VR, V sub R. V sub r is the resting transmembrane potential. VTM can change at whatever point. It's just the potential of the membrane, the transmembrane potential. At any point, VTR can equal V sub r, or VTM can be something else, depending upon what's happening. Okay? So V sub r, the resting VTM, it's just the transmembrane potential of an undisturbed cell. This number is negative 70 millivolts. So a millivolt is 1/1000th of a volt. Has anyone stuck their tongue in a 9-volt battery before? Yeah. Imagine that shock divided by 9,000. Not amazingly strong, but measurable, easily measurable. Okay? The transmembrane potential is minus 70 millivolts. And the fact that it's minus versus positive is just an arbitrary convention of how they build the patch clamp meters and what we decide to call negative. Where is the way we define the terminals on it, right? But we call it negative 70 millivolts. So the way we maintain this is through this protein right here. It's the sodium-potassium exchange pump. It's a protein that sends 3 sodium out and brings 2 potassium in. So what kind of transporter would that be called? It's an active one because it's using ATP. What else? What's another thing we can say about it? It's an antiporter. It's an active antiporter, sending 3 sodium out, 2 potassium in, with a net charge change of +1 to the outside. Remember, there's all this negative protein trapped inside the cell. So we're establishing a transmembrane potential. We're burning ATP to do that. We have to use energy to do it. But if we just kept doing it, this will go until the ATP runs out. And what happens when ATP runs out? You die, right? Or you go have lunch. One of the two. This will just keep going until you run out of ATP. So that will establish a transmembrane potential that's more and more extreme with time. We need some way to ameliorate that and keep it stable. So not only do we stick this thing in the membrane, but we also stick these leak channels. These 2 channels that are just constitutively open. Constitutively means all the time. All right? Constitutively open. Always showing up. And so the fact that it's at negative 70 millivolts is dependent upon what? I could give it to you, but I'm pushing to see how critically you can think. What do you think? Someone who doesn't talk to me. Somebody please talk to me. Nobody wants to talk to me. Guess. Dare. Dare to be wrong. Dare to be wrong. Okay. You talk to me.
The potentials of the
The potentials of them, right, okay. So that's part of it. But specifically, why negative 70? Yeah.
Is it the rate of the sodium-potassium exchange pump that determines it?
It's the rate of the sodium-potassium exchange pump with relation to what? You're getting there. We're going to design this membrane. How are we going to do that? So let's, if we're going to follow a recipe, what does the recipe say? Does the recipe say chuck a bunch of sodium-potassium exchange pumps? What? Yeah.
The number of the channels?
Not just the number. not just the number, but the ratio, the ratios, okay? The ratios of expression patterns. So this is how genetic control is going to dictate this, the transmembrane potential. It's the ratios of sodium-potassium exchange pump, which has intrinsic kinetics. It has intrinsic kinetics to the way it works, related to the availability of ATP, but also how many of these different sodium and potassium leak channels are you going to stick in so that stuff is able to go back at a rate that relatively matches the intrinsic kinetics of the transporter, the active antiporter, to give us the potential we want. You digging it now? Okay. So the sodium-potassium ATPase is a really cool protein. I've already sort of said all this, but this puts it in word form here. So it stabilizes the resting potential when the ratio of sodium entry to potassium loss through the leak channels is roughly 3 to 2. All right? When the concentration gradients of sodium and potassium on either side of the membrane allows leaks through the expression pattern of the leak channels so that sodium and potassium are leaking across the membrane relative to one another in a 3 to 2 ratio, that's when it matches the sodium-potassium ATPase. You follow? You follow that? It's just trying to get the expression patterns of the sodium and potassium leak channels such that the sodium and potassium are leaking across at a relative ratio of 3 sodium one direction to 2 potassium the other direction, which is matching what the sodium-potassium ATPase is doing. Okay? So when the system comes to that equilibrium, that's when we're at the resting potential. And this is what, and it turns out that potential that the cells like is negative 70 millivolts. All your neurons, all your muscle cells, so like the bulk of the cells in your body, are charged up. Takes one ATP. Without ATP, neurons stop functioning, muscle cells stop functioning, and you are toten. Woo! What happened there? What did I just do? Okay. Oops. All right. Oh, yeah, this slide. So we're going to put this together. We talked about electrical gradients. We've talked about chemical gradients. They work together. The forces of each of those two concepts work in concert to form the electrochemical gradient. For any given ion, be it sodium, potassium, what have you, the action that ion takes is the sum of these two forces, the chemical and the electrical forces that are acting on this ion. So this is a form of potential energy. It's a form of potential energy. So here's the chemical gradient. We have, and I apologize for the heteronormative analogy that I have here, but it works. So we have lots of males over on this side and none on this side of the room. Well, give them some time and everybody will spread out because everybody likes elbow room. Okay? So there's a gradient, a chemical gradient, sending them this way, driving them this way. So imagine this is an elevator or something. I don't know. Here is an electrochemical gradient. So you're assuming here that this is that the males are attracted to the females, right? So there's chemical gradients that are driving both either way, and an electrical gradient. So each blue figure wants to pair up with a black figure because they're attracted to one another. So at equilibrium, we have negated both the chemical forces and the electrical forces driving diffusion across this imaginary line, all right? This is the equilibrium potential, So let me think about, well, let's just keep moving. So transmembrane potential with no net movement of a particular ion across the plasma membrane is what we call the equilibrium potential. So different ions are going to have, different ionic species are going to have different equilibrium potentials. So for example, we'll get to it, examples in a moment, but I'll just say maybe sodium has 1 voltage at which that voltage is not going to cause sodium, there's going to be no net flux of sodium across the membrane. As many are moving in one direction as are moving in the other. But that potential may be different than it is for potassium. At that potential, some potassium may want to move in a net direction, whereas potassium has its own equilibrium potential at which for every potassium that goes one way, another will go in the opposite direction. All right? This is the notion of equilibrium potential. So at any given time, the membrane potential is going to be, the membrane is going to be driving towards a potential as determined by whatever ion is the most permeable to that membrane at the time. Okay? So let's say the membrane is at minus 60, and at minus 60, sodium really wants to go across the membrane, and its membrane potential, its equilibrium potential, is at plus 30, whereas potassium's is at minus 60. At minus 60, potassium is not moving across the membrane at all, and maybe the gates are closed, and it's not able to move. Sodium is able to move across the membrane, and so it's going to start driving that membrane potential towards the equilibrium potential of sodium. Whatever ion is most permeable at that moment, that membrane is going to be moving towards its equilibrium potential. Okay? So here we have potassium is able to move outside of the membrane. And so this membrane potential is going to be as close to the potassium equilibrium potential as it can be, because this is the one that's able to pass. Or sodium. Say sodium is able to pass. This transmembrane potential is going to be as close to the sodium equilibrium potential as possible. Right? And here's an example where they're both able to be freely passing, and it's going to be some membrane potential in between the two. Okay, so this is actually an important slide. This is an important slide. In the top panel, we're going to look at the electrochemical gradient for potassium. At minus 70, at V sub R, the potassium chemical gradient wants to drive the potassium outside, doesn't it? Why? Why? I mean, it's obvious, though. Just look at it. This isn't a trick question. Why does, in this picture, don't even worry about the 70 millivolts for a moment. In this picture, why is the chemical gradient of potassium heading in that direction? Just look at the picture. Yeah. Totally. Totally. There's a lot of potassium inside and not much outside. Why is that? Why do we have so much potassium inside? It's being pumped in by the sodium-potassium ATPase. Okay? So we've been burning ATP, pumping potassium in here. Its chemical gradient wants it to go outside. Its electrical gradient kind of wants it to come inside. Why is that? What's that? Bingo, thank you. Yeah, there's all this protein inside here that's negatively charged. So there is a negative 70 millivolt resting potential on that membrane. And so a little bit of the potassium wants to come in because of an electrical gradient. So the sum of these two forces gives us the net electrochemical gradient, right? The net electrochemical gradient for potassium wants to drive the potassium out of the cell. You see that? Mostly because of its chemical gradient. The electrical gradient is actually driving potassium in the opposite direction, so it mitigates the chemical gradient a little bit. This is at minus 70. All right. Let's change the electrical membrane potential. So now at the equilibrium potential for potassium, which is negative 90 millivolts, the chemical gradient is the same as the electrical gradient. Those forces are equal and opposite. All right, the forces are equal and opposite. There is the same concentration of potassium on the outside as, not the same. The concentration difference of potassium inside versus outside, that force, because of the chemical gradient, is equal and opposite to the electrical gradient driving it inside. All right? We've let some of the potassium out, and we've sent it down to minus 90 millivolts. These are real numbers. These are the actual numbers. So equilibrium potential for potassium is negative 90 millivolts. Let's look at sodium. Here is the resting transmembrane potential. Honestly, this slide is almost the whole lecture today in some ways. I mean, this is it right here. The sodium chemical gradient is the opposite of potassium. Chemical gradient wants to come inside. Why? Someone else. You. Yes, you. Why? Great. Exactly. Why? It's the same reason. I'll give you a clue. It's the same reason and the same answer as to the question of why potassium's chemical gradient was driving it that way. What was the answer to that question? Why is there so much potassium in the cell? Why is there so much sodium outside the cell? Yeah, you.
In the case of the potassium, it would be pumped in.
You got it.
So the sodium is being kept out.
Yeah, the sodium's getting pumped out actively. We're burning ATP. The sodium-potassium ATPase. We're burning ATP to stick it outside the cell. That's why there's so much sodium out here and there's not much in here. So that chemical gradient is saying, get inside. The electrical gradient is pointing in the same direction as the potassium gradient and the same strength, because honestly the charge on sodium is the same as the charge on potassium. So these two are actually reinforcing. At minus 70 millivolts, there's a huge electrochemical gradient telling sodium you want inside. You want inside the cell. They're adding up to one another. So at sodium's equilibrium potential, this is where the chemical gradient has been allowed to relieve itself such that it matches the electrical gradient. All right, so let's open some channels here. We're at minus 70. Let's open some channels. Sodium floods into the cell, floods into the cell. All right, disrupting this transmembrane potential because we're allowing charge to flow and we're going to change the electrical character of the membrane. So now it goes from minus 60 to plus 66 millivolts. We've actually flipped. It's like flipping the terminals of a battery. What used to be positive and the other was negative is now reversed. This terminal is now positive and the other is negative. This is what's happened in the membrane potential here. So the chemical gradient, there's still a little bit more outside than there is inside because remember we were pumping 3 sodium out for every 2 potassium that came in. There's a lot of sodium outside the cell, much more than in here. So there's still a little bit of a chemical gradient. Enough such that enough has flowed so that it actually flipped this electrical gradient so that it now matches and balances the chemical gradient. So this is equilibrium. No net sodium is going to pass. 1 sodium may come in for every sodium that goes out. All right? 1 sodium may come in because of the chemical gradient force, and 1 sodium may move out because of the electrical gradient force. It's the equilibrium potential. All right? So what we have here is a pretty nifty situation. We have 2 cations which have opposite polarity equilibrium potentials. Not only do we have a mousetrap, but we've designed a way for the mousetrap to reset itself. Okay? This is the spring on the mousetrap. This is how we reset the mousetrap. Okay? Let's see how that works. Any questions so far? Yeah. Here?
How we arrive to the net electrochemical gradient.
Okay, so there is a strong chemical gradient. You see that, right? There is a little bit of an electrical gradient. It's actually driving it in the opposite direction, right? Because there's more negative charge inside than there is outside. All right, and that's because there's a bunch of negative proteins in here, and there's a ton of sodium outside. So there's some charge separation. It's a pretty big charge separation. And so there's a little bit of an electrical gradient that wants to drive any positively charged thing inside. These 2 opposing forces add up. They, if there was... Yeah, I wanted to do this class flipped eventually. I tried it one year when I had pneumonia and make everybody watch the lectures online and then come in and we do games. So we get up and walk around and do games. There's an activity I could do where we could all get up and I could create a diffusion gradient, but then also have rules of attraction, like positive and negative, to try to demonstrate this. It's a fully just physical property of what's happening here with the rules of attraction and diffusion. There's this overwhelming force to drive things out because of it being crowded in here, the chemical gradient. And then there's a little bit of an electrical gradient driving it inside. So imagine I'm sitting in here and there's somebody else, my clone is outside. And me in here saying, okay, everybody, out the door, get out of here, loud voice. And then me, my clone in the hall, is saying, okay, head inside. Yes, yes, yeah. Yeah, there's more of a chemical gradient. Yeah, and so there's going to be a net force driving people out because I'm so loud in here. But some people will say, oh, yeah, he told me to go back in. And he like sneak back in. A few people will, right? So they're opposing forces. One's just much stronger than the other. And the smaller electrical gradient kind of mitigates this big force. And we get this net electrochemical gradient. All right? Yeah.
So because of the polarity of the electrochemical equilibrium,
Yeah.
I'm assuming that you would have to open one channel before the other.
Yeah, we're going to see it. You just hold on to your hat. It's coming. Yeah.
For the electrochemical gradient for sodium, I don't understand why both of the gradients are pushing the sodium. I get the first one and then the second.
So you got the chemical gradient, right?
Yeah.
Yeah. The electrical gradient is the same as the potassium. Potassium and sodium are both cations. And at minus 70, it's a little bit more negatively charged in here than it is out here. This is a little more positive, this is a little negative. Okay? As reflected here by we're measuring that, and we see at minus 70, it's a little bit more negative in here than it is out here. And sodium is a positive ion. So electrically, it's going to want to come in towards that negative charge, right? Just like the potassium. It just turns out that the chemical gradient and the electrical gradient for sodium are pointing in the same direction. But the chemical gradient for sodium is pointing in the opposite direction of the chemical gradient for potassium at the resting potential. Why? Anybody? Sarah, you tell me. You're clearly paying attention. You got it. You can do it.
Is it because that it wants to go to the less concentrated, make it more balanced that way, versus with potassium, if there's more on the inside, it wants to get more out?
If by saying that you mean it's because of the sodium-potassium ATPase, you are right. It is because the sodium ATPase is driving sodium out and potassium in. They're going to have opposite chemical gradients, right? So for every 3 we chuck outside, a sodium outside, we're bringing 2 potassium inside. They're just going to naturally have opposite chemical gradients. It's because the sodium ATPase is an antiporter that all of this is possible. It's because it's an antiporter that all of this is possible at all. All right? If it was a symporter, we'd have the spring for the trap, but we wouldn't be able to reset the trap ever. Because we're flipping the roles of 2 different cations, and we're causing a dissymmetry in them in terms of the established chemical gradients because of the antiporter that we've made. All right? The antiporter puts a bunch of potassium inside and a bunch of sodium outside. All right? Yeah.
Is there a reason why the channels specifically push the sodium outside and potassium inside?
You mean why did it evolve to put sodium outside and potassium inside? That's a good question. And I can only speculate as to the answer to that. But I would guess it's partially because these ions are playing other roles in the cell, and particularly potassium is playing other roles. And, it may be advantageous to have lots of potassium in the cell natively at the resting potential. I don't know. I have to think about that one. All right. Any other questions there? All right. So the membrane, the VTM varies with the permeability to sodium or potassium. And we're just going to come up with ways to vary the permeability to either of those ions to cause these membrane potential dynamics that are going to represent the graded potentials, the action potentials that we want. So it's important to have these leak channels. I've talked about those so that we can get the specific resting potential that we need. And we have these active channels, these gated channels that can open and close in response to stimuli. That stimuli can be some sort of potential. At V sub R, most channels are closed because they're chilling out. At V sub R, really all we need are the sodium-potassium ATPase and the leak channels, which are constitutively open. They're always open. They're not gated. They're just open. Okay? And that's helping us get minus 70. And then you change the potential, and these gates start to open. We're going to talk about how to begin to open to spring these traps. I already showed you, didn't I? We have chemically gated channels that, like the acetylcholine, opens up some of these gates. We have electrically gated channels. We have mechanically opened channels. Okay, chemically gated channels. Here we go. So this opens in the presence of specific chemicals. For example, acetylcholine at a binding site. These chemically gated channels are on the cell body of a neuron at the dendrites. Binds the acetylcholine, and the ion can pass. All right, voltage-gated channels are going to respond to a change in the voltage of the transmembrane potential. So here, wow, you've already seen this picture. I showed this to you several slides ago. Resting, it's closed. Change it a little bit. This opens. The ions flood across at a different potential. This gate closes. How does that happen? Well, it happens by having different alpha helices that have charge-carrying amino acids that at different potentials change their ionization state. Okay? They change their ionization state and either swing in towards the barrel because they're attracted to something here, or swing away from it because they've changed their ionization state, because the membrane potential has changed. All right? Voltage-gated proteins. Pretty cool, man. All right, mechanically, I already talked about this, but here's just a picture. It's pretty amazing how these work. It's like I should bring that in sometime. I have this beautiful origami this woman made for me that it slides open and closes. It's a pore, and this protein works quite the same way. You deform the membrane, and this pore opens up, and you release it, and it slides back closed again. All right, so here's just a couple definitions. Wow, I spent most of the class going over just resting membrane potential. We're going to get to greater potential now. Depolarization is a decrease in the transmembrane potential, so pushing it closer to zero. And hyperpolarization is moving it away from zero. Is that pretty straightforward? Okay. Graded potentials. Local potentials that do not spread very far from the site of stimulation. In fact, they get weaker as they move away. Any stimulus that opens a gated channel is going to produce a graded potential. All right, so let's go through this step by step. We have the resting state of the surface of the neuron. It's at minus 70 millivolts. Lots of sodium outside the cell. We're going to bind some sort of chemical. Let's call it acetylcholine. Who knows? Your favorite neurotransmitter. And it opens the chemically gated sodium channel. Sodium rushes in, floods the cell because that's the direction of its electrochemical gradient. Remember, the electrical gradient's pointing in that direction, and its chemical gradient because of the sodium-potassium ATPase. So that's stimulation. We've stimulated the membrane. And we notice now that the membrane potential starts to shift in response. We're releasing some of the pressure of that polarization. So it's going to move towards the equilibrium potential of sodium. Remember I said that the membrane potential is dictated by the ion with the greatest permeability to the membrane. All right, second step. This spreads out. We're going to get a local current. This was where the stimulus was, right in here. All this sodium starts building up here. Well, it doesn't want to just stay there. It's going to diffuse down its chemical gradient horizontally inside the cell. It spreads out, and it's going to change the membrane potential outside of the point of original stimulus. So here, this is continuing to depolarize in this area, or move towards the equilibrium potential for sodium. And so we are at minus 60, but as we move away from that point of initial contact, it's less depolarized. We call that local current. So here's gradient potential depolarization. It's a shift in the transmembrane potential towards 0 millivolts. So here we have here's minus 70 millivolts, here's 0 millivolts, and this is where on the membrane, so that you can imagine this as the surface of the membrane, and in this dimension we're measuring charge. This is where you stimulate it, and we depolarize the membrane right there. But as you move away from the site of stimulus, that depolarization becomes less and less. This was like my burping analogy that I've never used before. Seems like a burp is better than a fart, though. And then hyperpolarization. So this is what potassium does. Potassium's equilibrium potential is actually higher than the minus 70 resting potential. All right? So it's a shift in the transmembrane potential towards minus 90. So you can imagine it in the same way here. Here's minus 70, and instead of heading upwards towards 0, we're going to head down towards minus 90. And at this point of stimulus, that hyperpolarization gets less as you move away. So it increases the negativity of the resting potential. All right. So here's just a larger scale schematic. We have the initial point of stimulation here, and then that spreads out across the membrane. All right? The stronger the stimulus, the more acetylcholine you dump, or whatever neurotransmitter, the more of these chemically gated channels are going to open, and the more sodium is going to be able to flood in and cause a stronger and stronger depolarization, a stronger reaction. All right. So when the stimulus is removed, you're going to, like when the acetylcholine diffuses off of those channels, which it will, they'll get recycled, those channels will close, the membrane potential is going to return to the resting potential. Why? It's the same answer that you would have worked for several other questions that I've asked today. Yeah. There you go. The balance between the sodium potassium ATPase and the leak channels. That's it. All right. So here is some sodium stimulation, and it's depolarizing, and then we remove the stimulus, and then it repolarizes because of the sodium potassium ATPase. Here we're going to open some potassium channels with some kind of chemical, and it's going to go down here. We remove it, and it brings it back to normal. All right. So gradient potentials are all over the place. You see them at the motor end plate. This is where we release acetylcholine into the synaptic cleft. It's at the cell dendrites or the cell bodies. We have this exocytosis of the acetylcholine, which goes, and this is how we release the chemicals, the neurotransmitters, on to the chemically gated receptor channels. Okay? You guys follow me? This is foundational stuff right here. All right, so we've talked about graded potentials. Now let's get to the good stuff, the action potential. Large, rapid, reversible, propagated decrease in the transmembrane potential. It affects an entire excitable membrane, an entire membrane. It's the way that we can link graded potentials in the cell body to action at the synaptic terminals. All right? Excitable cells are going to have action potentials. Inexcitable cells do not. All right? Any cell is going to have a transmembrane potential that you can disrupt, transiently. But excitable cells are going to have action potentials. All right. So it's going to happen due to a sequence, a carefully balanced sequential increase in sodium, calcium, and potassium conductance. This is going to be triggered by reaching some sort of threshold by which one of these voltage-gated channels is going to open, and it's going to cause a rapid increase in sodium conductance. So first, with the graded potentials, we were talking about chemically gated channels, right? You put a neurotransmitter on, you open it up, and blah, blah, blah. Action potentials are going to be using voltage-gated channels. They are sensitive, so this is depolarization that's sensitive to depolarization. When a graded potential causes enough depolarization so that spreads into a part of the neuron, the axon hillock, that has these voltage-gated channels in it, then those channels are going to start opening. And as they open, they're going to open ones next to them. And it's just going to propagate along the whole length of that excitable membrane. Okay? You guys following that idea? So we had chemical gated channels for the graded potentials. And if we get a lot of those, we're going to deform, that stimulus will be strong enough that it will send a deformation in the resting membrane potential to the point where it gets to part of the membrane that has these voltage-gated channels on it, and then the trap is going to spring. All right, we'll see how it works. Here's one of these voltage-gated channels. Here's a little loop that has one of these four loops that are all have these ionizable amino acids on them that can swing open and close depending upon what the membrane potential is. And this is where tetrodotoxin binds for pufferfish. Does anybody ever eat sushi? Anyone had pufferfish? It's a little bit risky, but why would you want to eat such a nice little guy anyways? He's cute. This toxin that these smiley fellas produce goes in here and binds this and just leaves the gates open. So you go into a kind of tetanus where you're like, you can't do anything. It's a neurotoxin. Messes up these voltage-gated channels. So there are 4 steps to generate an action potential. Step 1, you've got to depolarize the membrane to threshold. That's by the summation of a bunch of action potentials somewhere else in the cell body. And that membrane potential has finally impinged upon the axon hillock where we have these voltage-gated channels. So we depolarize to a threshold. The threshold is going to turn out to be -60, -65, something like that, millivolts. And we're going to activate these sodium channels. These sodium channels are going to swing open. Sodium rushes in. Depolarizing the membrane. At a certain membrane potential, the sodium channels are going to be inactivated, and other voltage-gated channels that pass sodium are going to swing open at that potential. And sodium is going to begin to rush out, thus restoring the transmembrane potential and returning the cell to normal permeability. Okay, that's an overview. We're going to go through each of this step by step. All right, resting potential. We have sodium channels in purple, potassium channels in orange. We're going to depolarize. So here's a local current that's coming from some graded potential out in the cell body. It's going to deform our transmembrane potential until we get the threshold for this channel, this sodium channel, whatever that loop is, that ionizable loop in the protein, it's going to swing open when the transmembrane potential gets to minus 60. When that happens, these sodium are going to start coming in. This is the all or none principle. Once you spring the trap, it's all or none. We're going to light up the whole membrane. Graded potential, it's this gradation, right? It's this gradation. Some membrane is stronger stimulated or stronger depolarized than other. But here with this stuff, this is a mousetrap. A mousetrap doesn't swing halfway. It's all you flip the switch, the whole membrane's going to go. 2, we see the sodium start rolling in, flooding in, changing the transmembrane potential. We even flip the potential. At that point, when we get down to this inactivation threshold potential, the inactivation gate in the sodium slams shut. Sodium's done. And the gate for potassium opens up, and potassium rolls across its electrochemical gradient. All right? Remember, it's got a strong chemical gradient driving it outside because we've been pumping potassium inside with the sodium-potassium ATPase. So now it's going to start flooding outside, and that's going to drive the potential towards potassium's equilibrium potential, which was minus 90. Sodium's was plus 30, 35, something like that. Okay? Who feels lost? A little bit. None of you? A little bit? A little bit? What part is it is catching you up?
I just don't really get what it means. I understand the mechanics of it, but why does this happen in the cell? What does it lead to?
You mean why are we going through all this?
Yeah.
Yeah. We're trying to send a signal to another cell. We're trying to send a signal to another cell. And then that signal has informational content. It has meaning that is imbued with some sort of representational content in the brain. But right now we're just trying to talk about the mechanics of how that signal is being sent. All right? Does that answer that question? A little bit? I mean, let me get to the fun stuff, the information processing. But this is, yeah.
Why is the active different? Why does the whole membrane go with active?
Yeah, because a chemical gated channel is for a graded potential, right? And you have to be near the site of that chemical. That chemical's got to be around for those channels to be open. It doesn't really, like, the transmembrane potential is not what is determining the permeability in terms of a graded potential or a chemically gated channels. But here, they're voltage-gated channels. And so when the membrane is having a deformation in the transmembrane potential, it's like a positive feedback thing. You know, it starts to throw open more channels near it, and it spreads, and it spreads, and it goes over the whole membrane. Does that make sense? Yeah. So that's one of the distinctions between a graded and a voltage, a graded and an action potential. Okay. Step 4, we return ourselves to normal permeability. So at minus 90 millivolts, this door swings shut on the potassium gate. And The membrane is a little bit hyperpolarized now at negative 90 millivolts as these potassium channels finish closing. So the transmembrane potential is now going to return to minus 70 from minus 90 through the action of the sodium-potassium ATPase in the leak channels. Okay? Yeah?
So if you could draw a graph of the voltage across the membrane as like the voltage as a function of time,
Yeah.
would it be like a sine curve?
You'll see. I got one here, if I can get to it. Right there. Is that what you're talking about? That's it. So here's threshold. Here's resting membrane potential. So there's some graded potentials, chemically gated graded potentials out in the cell body. All right? All these incoming signals that are getting added up. Some are, we won't get into the information processing yet. Hopefully I'll get to it. I probably won't. But we reach a threshold, and suddenly these sodium channels swing open. We depolarize. The voltage swings way up here, and then the sodium channels shut, the potassium channels open, and the potassium begins to flow towards its equilibrium potential, which we said was down here at minus 90, right? So now we're going to repolarize. The sodium channels, the voltage-gated sodium channels are the spring of the trap, and the potassium ones are how we reset the trap. All right? So we get back down here, and the sodium-potassium ATPase brings us back to our normal resting potential. So there's this notion of an absolute refractory period. Once you spring the trap, it's happening. You've committed to it. You can't stop it. You can't make it less than it was. You can't make it more than it was. It's happening. So it doesn't matter if downstream over here, there's a lot more, all of a sudden there's a lot more graded potentials coming in. You're not going to be able to spring the trap twice during this point, this refractory period. You guys follow that? What refractory means, if someone's refractory, they're not doing what you want them to do. They're not doing what they're supposed to do. The neuron, you can't spring the trap because it's already been sprung, and we're going through this process. We're committed to it. We have to wait till it's over. So then there's this relative refractory period right here, where it's closed. We're kind of near normal. If you have a really strong response, you can kind of get it to go again right here before we're completely back to normal. But it's hard. You got to have a lot of strong stimulus. The relative refractory period. Absolute refractory period, nothing can happen. Relative, you need a strong stimulus. So here is sodium conductance. This is the same voltage curve, and here you see in blue and green how much potassium and sodium is actually traveling. So this is like numbers of sodium and potassium ions traveling. Okay. So here we get the stimulus right here. We open the gate, we reach threshold, sodium starts flooding in, and the gate closes. This shouldn't be slow. This should probably be a precipitous thing right there because everything drops off right away, right? And sodium comes down to zero. And then this is where the potassium gates open. Potassium floods across the cell until its gates start to slowly close, and this should actually probably be more like that as well. All right? So here it's all the same stuff, just a different way of depicting it. Different cartoons. I want to get to some other concepts. I'm going to let you look at that if you need to reinforce that concept in your head. But this is just, it's the same. It's redundant. All right. So there's this temporal profile, and I've been showing this. This is how the change in transmembrane with time. You learned the word temporal yesterday, right? With time. But you can also think about it in terms of spatial action potential profile. Here's the axon. And so at a given point, you've depolarized the membrane, and that's spreading away from the point of stimulation at the axon hillock, and it's spreading down the axon. Behind it, there's this wave of repolarization. You follow that? So there's this spatial wave of propagation, of depolarization that's moving down the length of the axon towards the synapse. All right, so here is a different way of looking at it. Here's an axon. We've depolarized here. This is an action potential in progress. It had been right here, the slide before, for example, but this is repolarizing. This is the refractory, relative refractory period, and this has already gone through action potential and is back at normal. So this wave is going to start moving this way down the axon. You're maybe asking, well, why doesn't it head the other direction? Because it all started here. There is no electrically gated channels back this part in the cell body. All the electrically gated channels are this way. And this is where the trigger is. And it sets off the trigger here, and it just moves this way down the axon. Yeah?
Is the threshold voltage for the inactivation of the sodium channel the same as the threshold for the activation of the...
Almost. They're very close, yeah. Yeah. In fact, they may be identical. They may be the same, it may be the same loops of proteins that got mixed in those genes. It may be the same genetic sequence. I don't know, though. It's a good question. I'm not exactly sure about that part of it. So, two methods of propagating axons. Continuous propagation. So this is kind of important, sort of. Continuous propagation is it's just rolling down the surface of a membrane. And so you have this real-time flow of ions and depolarization events that happens. Okay. Or, so we're kind of watching it happen here. There's this local current. It's shifting down the length of the axon, and we're just watching how it affects the membrane dynamics. The second concept is saltatory propagation. And this is what happens in most of your cells. I'm assuming nobody in here is a giant octopus who use the continuous propagation. So saltatory propagation uses myelin. Myelin is just insulation on the axon. It helps, what it really does is it prevents electric field leakage around that wire. So any wire is going to give off an electric field that has current passing through it, right? This is why you take a nail, you wrap wire around it, put a battery on it, and it turns it into a magnet, right? Because of electric field leakage out of the wire. But you insulate it here, you prevent electric field leakage, and you can get a much quicker jump of electrical potential from this point to this point. In between, these are each called nodes. Nodes. And so at a node is where you have electric gated channels, but in between, you don't need them. This is an internode. You have a stimulation here, and it's going to jump quickly to the next node. And then it gets boosted again and jumps to the next node. It's just like in the electrical grid, you see those transformers on the power grid, on the electric lines. The electricity is coming through the wire, and you start to lose, with time, you start to lose power, and then they boost it up again, and it keeps going. That's what the transformers are doing, right? It's the same thing here. We have this insulation in the form of myelin, and we'll talk about that in the lab a little bit more. So it's faster, uses less energy. All right, saltatory propagation. Let's watch how it happens here. So we have depolarization at the first internode, or maybe the axon hillock. We have a local current that jumps quickly to the next node, depolarize there, and then jump to the next one quite quickly. And it propagates down the length of the axon. So we see it. Here's another, again, all redundant. Just jumping from one node to the next in this saltatory propagation. This is such a fun concept. I love it, but I'm not really going to get a chance to talk about it in great depth. It's the notion of signal processing and analog versus digital, or one way to think of it is FM and AM signal propagation. So gradient potentials encode information in terms of the strength of the stimulus, their amplitude. Right, the more stimulus you dump on, the stronger the response. Or action potential encoding is by the frequency. It's all or none. It's always the same intensity. And if you want to send a stronger signal, you don't send a stronger signal. You send a more frequent signal or a less frequent signal. This is like frequency modulation in FM radio. And this is like amplitude modulation in AM radio. This is the difference between AM and FM radio. But your neuron takes advantage of both ways of encoding information. So gradient potentials are amplitude modulated, and action potentials are frequency modulated. It's a little bit of a nuance for the nerds out there. Okay, so here's a list comparing the two of them. I've already gone through all this. You can look at it. I do want you to understand that. I want to flash through it. The concept here, don't be afraid of all this math. It's just that the larger the diameter of the neuron, the faster the propagation. So small diameter is slow propagation. It's just because of resistance. It's just like you want really thick speaker wire for your speakers. All right. Yeah, and this kind of goes through why myelination and diameter both are useful. Disregard this. So disregard this too. All right. We have an action potential arrive at the synaptic terminal. It's going to cause degranulation or release of the neurotransmitter to go across the synapse. We have an action potential that releases releases a little bit of neurotransmitter, or one that releases a lot of neurotransmitter, but there's only a few receptors that are going to get opened. Here's a synapse that has just a little bit of neurotransmitter, but a lot of receptors. Still no action potential coming out of this. Here is an example of lots of neurotransmitters dumped on lots of receptors. So we have lots of gradient potentials. So now we're able to stimulate an action potential that's going to move downstream. You follow that? So there's a connection between the amount of gradient potentials that are created and whether there's an action potential that happens. The receptors dictate the effect of the neurotransmitter. Okay, so these classes of neurotransmitters, I unfortunately am not going to get to talk about too much, but I'll make this point. Excitatory and inhibitory. There are some that depolarize the membrane, and there's some that are going to cause hyperpolarization and inhibit that neuron from having an action potential. You guys follow that? So excitatory and inhibitory neurotransmitters. And there's these different classes of them. All you need to know is just hear their names: norepinephrine, dopamine, for example. This is involved in Parkinson's disease, and and in cocaine use throws off your dopaminergic receptors. Serotonin, this is going to affect your attention and your emotional state. Serotonin reuptake inhibitors are real popular. I'd be surprised if someone out there isn't taking one of those. And GABA from glutamate. This is an inhibitory neurotransmitter that's used in muscles oftentimes. This stuff should all go out. I'm sorry. Yeah, G proteins. I wanted to talk about it. I always struggle. I hate rushing. But so I'm going to cut out all this neurotransmitter stuff. Spatial variation. There's a lot of stuff I didn't get to. Well, we'll just talk about the pathology. All right. Cocaine addiction and Alzheimer's. They have opposite effect on dendrites. So here is a normal dendrite. It's these dendritic spines that have the receptors for the synaptic glutons to excite and cause graded potentials that are going to go through the cell body and cause a action potential. If you are unfortunate enough to be a cocaine user, it's going to enlarge your dendritic spines. So your cells are hypersensitive. You've been hypersensitized. You are actually fundamentally changing the morphology of the neurons in the brain. And similarly, someone with Parkinson's disease has the opposite happen because of their pathology. They are having a withering away of these dendritic spines, so their ability to respond, their neurons' ability to respond has become lessened. So sort of opposite maladies, two different maladies with opposite neuronal manifestations. That's that. Okay. Are there any questions? Sorry, I spent too long trying to make sure the membrane potential dynamics sunk in. I didn't get to talk about all the neurotransmitters the way I wanted to. It's fun stuff, but it's there if you want to look at it. All right, take a break. See you in lab. Everybody feeling okay? Stunned? I'll tell you this. The first week of class is by far the hardest. Next week is the second hardest, and then it gets much easier. I throw the bulk of the material at you in this week and a half. You guys are already through a huge amount of material. And I do that so you have the baseline of understanding so we can work on these projects later in the semester. It's not going to be this hard the whole semester, okay? So take heart. Don't be stressed. Be excited. You're learning. You're young. You're smart. You're joyful.